Good afternoon. Today we are discussing the black tooth genetics and DNA and the genetic code. Actually, DNA and RNA is the both are under comes the nucleic acids. Okay, nucleic acids. Nucleic acid was first discovered by the Frederick Fisher from the nuclei of first cells, the leukocytes. From the discovered the surgical bandage and the called is the nuclei. The first one, Frederick Fisher, he discovered the nu nucleic acid, which cells is the first cells. Okay, pass it. Next one, nucleon was the later shown to be the mixture of basic protein and the phosphorus containing organic acid is now called as nucleic acid. Nucleon was the mixture of basic protein and phosphorus. Basic proteins plus phosphorus containing the organic acid is now is also called as nucleic acids. How many types of the nucleic? There are the two types of the nucleic acid. Polynucleotides. Poly mean many nucleotides. Two. What are the nucleic acid? Ribonucleic acid. RNA. And next one is the deoxyribonucleic acid. DNA. The both. These are the two types of the nucleic acid. Nucleotide. What is the meaning of the nucleotide? The monomeric units of nucleic acids are called nucleotides. Monomeric. The single monomeric units of the nucleic acid is also called as nucleotide. Nucleic acid there are also called as the polynucleotides. Poly mean many. Polynucleotide. Nucleic acids are also called as the polynucleotides. Nucleotides are the phosphate esters of nucleosides and made of the three components at a base of the as nitrogen atom, nitrogenous base. The 5 carbon is the sugar. Mainly are the nucleic acids. Is the nucleotides are the phosphate esters nucleosides and made of the three components is the mainly nitrogen atom. The 5 carbon atom is the phosphoric acid. Okay. Okay. Nitrogen bases. Nitrogens are the heterocyclic planar and the relatively water insoluble aromatic one. These are the nitrogen bases are the water insoluble aromatic components. Water insoluble aromatic molecules. There are the two general types of the nitrogen bases, both RNA and DNA, pyramids and purines. Pyramids and purines. What are the pyramids and what are the pyramids? Purine. Purines are the two different nitrogen bases with the furan ring composed the one is found in DNA. The two common furan bases are the DNA and RNA is the adenosine, adenine and guanine. AG. Adenine and guanine are the purines. AG, adenine and guanine is the purines. Next one is the adenine as the amino group on the sixth position of the ring. The sixth position of the guanine as the amino group C2 position of the carbonyl group at the sixth position. The adenine is the mainly the amino group are there contain the sixth carbon position, adenine, and the guanine also carbonyl group also present in the C2 carbon position. AG is the purines. Next one is the purines is over. One minute. Okay, nitrogen is purines and okay, purines is over. Next one is the pyrimidines. AG is the adenine and the guanine both are the purines. Actually, the sixth position of the adenine, they are containing the amino group. The second position of the carbon, they are containing the carboamino group in uh, guanine. Okay, next one is the pyrimidines. What are the pyrimidines? Thymine and cytosine. The thymine and cytosine are the both are the pyrimidines. Thymine and the cytosine both are the pyrimidines. The thymine, especially cytosine, the instead of cytosine, thymine and okay, the RNA they are containing the acid. Thymine contains the methyl group is the at the C5 position is the carboyl group C4 and C2 position. Cysteine consists of the hydrogen atom at the 5 position. Okay, amino group. And next one, the uracil usually there is no found in the DNA. This is the component of RNA. Okay. Yeah, actually, instead of thymine, the uracil present in the an RNA. Okay, RNA, especially uracil are the pyrimidine. These are the present in only RNA. The thymine, cytosine, both are they're present in the in DNA. Okay, both are the pyrimids. Instead of thymine in RNA, they are containing the uracil. Okay, it is the uracil component. Purine and pyrimidine, the both are the okay. The sugars. The next one is the naturally occurring the nucleic acid. Have two types of the sugars, the ribose and the dehydrose sugars, the dehydroxyribose sugars. Ribose sugars are found in RNA. It is the 5 carbon monosaccharides with the hydroxyl group, OH group on each carbon. Deoxyribose sugars found in the DNA. Actually, only ribose nucleus or ribose sugars are present in the RNA. Deoxyribose sugars are present in DNA. Okay, DNA deoxyribose is found in DNA. This is the 5 carbon monosaccharide lacking one oxygen atom at the second position. The hydroxyl group at the second position of ribose sugar is replaced by the hydrogen. The carbon atom of the ribose and deoxyribose present in the nucleic acids and nucleotides are designed with the prime mark the distinguished characters. Unprimed numbers are the atoms, nitrogen bases. These are the sugars. These are the diagrams of the sugars. Deoxyribose, D-ribose, and deoxyribose. D-ribose is the RNA. Okay. And the deoxyribose mainly these are the deoxyribose, the OH group reduction of the removal of the oxygen and reduction of the hydrogen, and they can form to the deoxyribose DNA. These are the sugars. Okay, sugar packer. What is the pentose sugar is the non-planar. This non-planarity and the termed as the puckering. Pentose ring can be the puckered with the two basic uh, puckered in the two basic conformations envelope and the twisted. In the envelope will form the 4 carbons pentose sugar are nearly coplanar and the fifth position of away from the prime. In twisted from the three atoms of the coplanar and the other two line away on the opposite sides of this plan. Twisting the C2 and C3 carbons related to the other atoms results in twisted forms of the sugar ring. Okay, these are the sugar packers. What is the meaning of the nucleoside? Nucleoside. Nucleotide mean sugars plus pentose phosphate group plus nitrogen bases is equal to nucleotide. Only sugar plus nitrogen bases giant form nucleoside. The bond between the sugar and the base of the called N-glycosidic bond. Okay, the phosphorus is the totally absent in nucleoside. Sugar plus nitrogen bases is equal to can form the nucleoside. The nitrogen base of the lies above the plane of the sugar when the structure is written in the standard orientation. That is the configuration of N-glycosidic linkage is beta group. and glycosidic linkage is beta group. The base is free of rotate around the glycosidic bond. Due to the rotation of the glycosidic bond, two different conformation are possibly. The two standard conformations of the base around glycosidic bond, glycosidic bond, the bond of the synonyms and the antonyms, anti-circular and the pyrimidines tend to adopt the anti-conformation almost exclusively because of the steric interference between the O2 and C5 in the synonym conformation, whereas the purine is able to assume both the forms. Okay, nucleoside is over. These are the nucleosides. The totally opposite in phosphate groups. Okay, totally absent in phosphate group, especially they're containing the nucleoside. So see, this one is the sugars and sugar, and next one is the nitrogen bases. Sugar and nitrogen bases, the nucleoside. Nucleosides are the nucleoside. Purine, the purine, antipurine, the synapurine, the antipurine, nucleotides. What is the meaning of the nucleotides? The nucleate are the phosphoric acid esters of nucleosides. The phosphate position C5 and nucleate can there is also called as the nucleotide 5 phosphate. Especially nucleotide mean nitrogen bases plus phosphate plus sugar equal to nucleotide. Okay, so this is the fifth position. C5 position are the phosphoric acid esters are formed. The nucleotide can have one, two, or three phosphate groups at the C5 position and are the called nucleoside, monophosphates, diphosphates, and triphosphates. The nucleotide, the nucleotides, structure of the nucleotides. What are the functions of the nucleotides? The primarily responsible for the formation of polynucleotides. What are the polynucleotides? DNA and RNA is the polynucleotides. Nucleate is the DNA, RNA. Polynuclear are the informational macromolecules. They store in the translate the genetic information. However, they also are required for the numerous other important functions within the cell. The functions are the formation of energy currency like ATP and GDP. Nucleotides act as the precursor of the several important coenzymes such as the NAD plus, NADP plus, FAD, and coenzyme A, serving as the precursor of the secondary messenger like cyclic AMP or cyclic GMP. ATP is a chemical link between the catabolism and anabolism. Especially ATP and NADP both are the assimilation powers. These are the mainly they can form the in photosystem and non-cyclic and cyclic photosystem. Okay, that is the chemical link between the catabolism and anabolism. Photosynthesis and respiration. These are the main chemical link between the catabolism and anabolism. It is the energy currency of the living cell. It acts as the donor high energy phosphate. ATP consists of the adenosine motifs, which is the three phosphoryl groups, the sequentially linked via phosphoester bonds followed by the two phosphodihydrate bonds. Referred to the high energy bond, the active form of ATP is the usually complex of ATP with the Mg plus 2 and Mn plus 2. What are the structure of the ATP? Okay, that is the structure of the ATP. AMP plus phosphate group, they can form the ADP. So these are the structure of ATP. Naming nucleosides and the nucleotides. Adenine, guanine, cytosine, and uracil, thymine. Instead of the thymine, they can form the uracil. Instead of the thymine, the uracil is present. Nucleoside, RNA, DNA. Nucleotides, RNA, DNA, nucleotide monophosphate AMP pathway, nucleoside diphosphate ADP, nucleotide triphosphate ATP. These are the sequences. polynucleate. Already we discussed the polynucleate. The polymers of the nucleotides which has formed to the condensation of nucleotides. The condensation reaction occurs between the alcohol of a 5-phosphate and one nucleotide, the 3-hydroxyl of the second, with the elimination of the hydrogen molecules, water molecules. The linkage between the two nucleotides is known as the phosphodiester bond. Mainly is that the linkage between the two nucleotides is also two nucleotides known as the phosphodiester bonds. Each nucleic acid is nucleotide that has been incorporated into the polynucleotide is known as a nucleotide residue. The phosphates of the polynucleates are the acidic. So at physiological pH and the polynucleates are the polyaminations. All nucleates are the polynucleate chain have the same relative orientation. The formation of the phosphodiester bonds in DNA and RNA exhibit the directionally. The primary structure of DNA and RNA linear proceeds in the 5' to 3' direction. The nucleotide sequence from the left to right synonym is the 5' to 3' direction. 3' to 5' direction. The order of the nucleotides occur in the polynucleate describes the primary structure by conversion of the sequence of nucleic residues, the nucleic acids, written left to right from the 5 end to 3 end. The interchain of the base pairing of the complementary nucleotide gives the secondary structure of nucleotide. Three arrangements. Okay, sorry, polynucleotides. The orders which is the nucleotides occur in the polynucleoside describes the primary structure, a conventional sequence of the nucleotides residues in the nucleic acid in left to right, right to left, the 5 prime to 3 prime, 3 prime to 5 prime. Interchain basically in the complementary nucleides gives the secondary structure of nucleoside. The three-dimensional arrangement nucleosides, that is the arrangement of the nucleic molecule, the space refers to tertiary structure of the nucleoside, that are the polynucleotides. Okay, the okay, next one. Already we discussed the nucleic acid, ribose nucleic acid, and deoxyribonucleic acid, and we discussed the deoxyribonucleic acid. What are the important features of the DNA? Mainly is that the quantitative estimation of the DNA, the various organisms will do the following some of the features. The first one is the genome is a single copy of genetic information of the cell. A genome is a single copy of the genetic information of the cell. Next one, next, the coded information within the genome is used to control the synthesis of the protein. The coded the information within the genome is used to control synthesis of proteins. The sequence of the genomes determines the sequence of amino acids in proteins. Gene is a segment of DNA molecules. Gene is a segment in DNA molecule. Okay, gene is a segment of DNA molecule. Next one, alleles are the different forms of the same gene. These are the quantitative estimation. And the next, another one is the amount base of the composition. DNA is the constant for the organism. The various different species. The DNA is called deoxyribose nucleic acid. Deoxyribose nucleic acid. DNA is the polynucleotide. Made of the many units is called the nucleotide. DNA is the macromolecules that due to the high molecular weight, especially the high molecular weight, DNA is the high molecular weight. DNA is the hereditary substance. The exactly DNA and the RNA is the both are the hereditary substances. DNA is the life molecule. These are the features of the important features of the DNA. What is the chemistry of DNA? Mainly, the proteins which are the linked with the DNA in chromosomes are known as the basic proteins or histones. Main, the proteins which are the linked with the DNA in a chromosomes are known as a basic protein or histones. Mainly, the rich amino acids, lysine and the arginine have the net positive charge. On hydrolysis of three molecules yield nucleotides. Each nucleotide is composed of the four nitrogen bases. Already discussed pentose sugar, deoxycarboxy, and the phosphate. Nitrogen base are the two types. One is the pyrimidines, and next one is the uh, uh, purines and the pyrimidines. Already we discussed the purines are the adenine and guanine is the purines, and cytosine and thymine is the both are the pyrimidines. Okay, and especially the DNA also they are containing the purines and pyrimidines. Instead of in pyrimidines, cytosine and thymine. Instead of thymine, this is present in the uracil in RNAs. The RNA is the single standard. Some most of the single standard, some exemption condition also double standard also seen to the in RNA. Okay, and next one is the molecules on pyrimidine base pairs with the pyrimidine base A equal to T and G equal to C. According to Chargaff law. A equal to T. The two bonds are present in the A mean adenine and thymine. One is the purine, another one is the pyrimidine. Okay. Uh, next one is the guanine and cytosine. G equal to C. Okay. These are the A equal to T and the G equal to C. These are the formula. In DNA, number of molecules guanine is equal to the number of molecule of cytosine. G equal to C. In DNA, a number of molecules of guanine is equal to number of molecules of cytosine. And next one, a number of molecules of adenine is always equal to the thymine. A equal to T. According to Chargaff law, A plus T equal to G plus T. Okay, A plus C equal to G plus T. A is the adenine and cytosine. A equal to T and G equal to C. Okay, these are the basic principle of the composition of the adenine and guanine. The bases are attached to the deoxyribose at the first carbon atom, where the phosphate is attached to the 5 carbon atom. Phosphate especially attached to the, say, diagram, page number 85. The phosphate also attached to the 5 carbon molecules. Phosphate, mainly they can attach the sugar molecules. Base, sugar, phosphate is also called as nucleotide. Is also called as nucleotide. Okay, sorry for the right. Okay, the base of the purines and the pyrimidines are attached to deoxyribose at the first carbon atom. The phos whereas the phosphate attached to the 5 carbon atom. Phosphate group is bonded to the 5 carbon atom. One sugar is covalently bonded to the 3 carbon of the next. Two such DNA strands are twisted around the other, just like the spiral staircase. Okay, that the DNA molecule. The individual polynucleotide chain exhibits the distinct polarity with 5 prime to 3 prime ends. The base of the pairs are the properly fixed in the double helix so that maximum hydrogen bonds can be formed. To active this, the two chains run to in opposite direction. Steering from the top molecule chain will run to the 5 direction to 3 direction, 5 to 3 prime and 3 to 3 prime to 3 prime. These are the important DNA. The next one, Watson and Crick model. DNA also discovered by the Watson and Crick. The two polynucleate chains of the primary metal from the double helix. The diameter of the helix is the 20 angstrom. And next one is the complete turn of the make complete turn is the 34 angstrom. Diameter the helix 20 angstrom. The each chain makes the complete turnover 34 angstrom. The internucleated distance is the one base pair to another base pair is the 3.4 angstroms. The sugar of the nucleotide is joined by the phosphate group to the sugar that just nucleotide. And there are the 4 main nucleotides, each containing of the deoxyribose residue, a phosphate group, and purine of the pyrimidine base. The molecular structure of DNA was proposed by Watson and Crick. This got the Nobel Prize in 1953. Franklin and Wilkins confirmed the structure of X-ray differentiation method. Mainly, Watson and Crick model is they can discover the DNA. The Franklin and Wilkins are confirmed the structure of by X-ray differentiation method. DNA is the discovered by the X-ray differentiation method. There is a wide variation in the base ratio, the AT and GC continents of the different DNA molecules. DNA is the higher plants and animals have the excess of a uh, adenine and thymine over guanine and cytosine. Actually, ATGCs are also they're containing the higher plants and animals, higher plants and animals. The viruses, bacteria, and lower plants are the rich in both AT and GC. Okay, mainly the higher plants and animals have the excess of the AT and GC. The virus are the and lower plants also rich both AT and GC. The basis ratios, the taxonomically related organisms are similarly not at random. The gene differentiation between the plants and animals due to the variation in the basis ratio. The DNA molecules with the high GC are more resistant to the heat than A plus T in the rich molecule. Heat denatures of DNA and the complementary strands separated from each other due to the breaking of their hydrogen bond. Every time the cell divides, the must make complete copy of the all genes. The products of the division, the two daughter cells, each receive the full complementary of the biological information posted by the parent. Dividing the cell has to carry the out of DNA replication. Replication of the DNA. Next topic is the replication. Watson and Crick model suggested the DNA replicate by the unwinding of the double helix. And with each strand of the acting as the template, each strand actually DNA is the double helix. Okay, double stranded model. The each strand they can act as a template. These are the template of the they can form the new template. This model is the replication is from semi-conservative method. They be semi-conservative. It shows the converted by the to be correct by the Matthew Melson and the Franklin Stahl using the technique known as the density gradient centrifugation. Mainly is the you can use the centrifugation method, separation by the centrifugation, semi-conservative method. Melson and the Stahl mainly transferred the E. coli bacteria growing the heavy metal nitrogen 15 isotope. DNA is the medium of containing only 14. The new DNA replication The new DNA replicated is the 14 and 14 nitrogen medium was intermediate density between the light 14 and heavy 15 and the DNA because replication was semi-conservate. The ratio hybrid of DNA 14 and DNA varied during the second their generation 1:1 or 3:1 respectively. The cesium chloride solution having the more or less equal density with the DNA is spun in the ultra centrifuge. The density of the strands was determined using the technique of density gradient centrifugation. The semi-conservate method of replication was verified photographically by J. Carrier in 1963. The technique is the autoradiography. Autoradiography mainly separation of the molecules. Okay, Melsen's stall mainly they can use for the nitrogen isotope N14 isotope, and the Melson and the Franklin Charles is using the technique of the semi-conservative method. Okay, next, a DNA replicates by the unwinding of the double helix with each strand acting as the template new double helix. The replication proceeds in the biodirectional manner. Replicating the biodirectional manner. The prokaryotic chromosome the circular with the single initiation point of the replication, and the eukaryotic DNA linear. Eukaryotic is the DNA is the linear and prokaryote is the circular. These are the major difference between the prokaryote DNA and the eukaryote DNA. The prokaryote DNA is the circular and eukaryote DNA is the linear. Eukaryote DNA is the linear with multiple initiation points of the replication. Only single initiation is seen to the in prokaryotic DNA. In eukaryotic DNA, there is the multiple initiation points of the replication. In bacteria, the circular chromosomes does not appear to the broken in the process of the DNA replication. An intermediate structure form, the DNA is unwound at the point and replication proceeds at the Y junction of the semi-conserved manner with the both the directions. The 5' to 3', 3' to 5'. Okay. Some of the Okazaki fragments also they can use in the semi-conservate method. The enzyme of the DNA polymerase involved in the DNA replication. There are three DNA polymerase also involved in the DNA replication. Polymerase 1, polymerase 2, polymerase 3 enzymes. These are the discovered by the Korenberg. Who discovered by the DNA polymerase enzyme? Korenberg. That's very important. They can ask into some of the complex examination point of view. Polymerase 1 is involved in the DNA repair. Polymerase 1 is involved in the DNA repair if mismatching as the at the standard. Mainly polymerase 1, polymerase 2, polymerase 3 is discovered by the Korenberg. The polymerase 1 mainly they can use for the DNA repair. The polymerase 2 also active replicating of the enzyme. Mainly polymerase 3, polymerase 3 also active replicating of the enzyme and not much known about the polymerase 2. Polymerase 3 is the active replicating of the enzymes. Okay. The enzyme helicase unwinds DNA at the site of the replication, and RNA polymerase creates the primers, 20 to 60 nucleotides in the length, depending on the space of the prokaryotes or eukaryotes. At the site of the Okazaki fragment initiation, the RNA primer provides the free 3 OH group. Okazaki fragments, continuous replication, the discontinuous replication leading. Let's see, page number 88. The DNA primer, the primer provides the free 3 OH group at the DNA polymerase 3 needs in the order of the synthesis of the Okazaki fragments. DNA polymerase enzyme adds nucleotides only 5' to 3' direction. DNA polymerase also enzyme adds to the nucleotide 5' to 3'. One strand or the present parent DNA, the lead strand is the synthesized continuously. The lagging strand has to be replicated in section. Short fragment DNA between 100 to 1000 nucleotide in length are also called as Okazaki fragment. What is the meaning of the Okazaki? Okazaki mean one strand or the parent DNA lead strand synthesized continuously. The lagging strand has to be replicated in section. The short fragment of the DNA between 100 to 1000 nucleotide length is also called as Okazaki fragments. Short fragment of the DNA between 100 and 1000 nucleotide length are also called as the Okazaki fragments. Named after the person is isolated, this is the fragment. It's joined by the enzyme ligase enzyme. That the enzyme ligase enzyme. DNA polymerase 1 removes the previous RNA primer and replaces the DNA nucleases. In eukaryotes and E. coli, DNA polymerase is also known, which is involved in the DNA replication. In especially in eukaryotes. In prokaryotes, eukaryote is less known about the eukaryote system. Okay, DNA replication and mainly genetic recombination of the bacteria in bacteria. These are the one type of the sexual reproduction in bacteria. Genetic recombination of bacteria. These are the bacteria and viruses have entered into the modern era of the molecular genetics. Okay, bacteria and the viruses the enter into the modern era of the molecular genetics. Since the bacteria and virus have the much less genetic material than the eukaryotes, the organization of this material is simpler. Millions of bacteria can be handled the single culture with the minimal effort. They will exploit the genetic studies. A genetic exchange of the bacteria takes place by one-way transfer of DNA from donor cell to recipient cell. There are the three ways of the achieving transfer of the DNA bacteria. There are the namely transformation, conjugation, transduction. In this process, DNA transfer from the donor bacteria to recipient bacteria because only is the manner is the DNA transfer that the bacteria DNA can move from one cell to another was established by the experiment Griffith 1928 and the Avril 1944 only complement bacteria can take up the DNA. And not all the cells are the component of transformed to be the component to be transformed. A cell must have the surface protein competence factor which binds to the extracellular DNA. When the extracellular DNA is brought into the cell, one standard to hydrolyzed by the DNA degrading enzyme DNA. The single strands brought into the cell is incorporated to the host genome by the form of the crossing over. Unlike the eukaryotic crossing over is not recipient. The conjugation, mainly conjugation. Conjugation is developed by the Ledenberg, Edward, uh, Ledenberg, Edward L. Tatum discovered the E. coli in 1946. Conjugation. Mainly, conjugation can exchange of the genetic material through the process of the conjugation. Conjugation mainly mm, genetic material come to the contact recombinant during the process of the conjugation. The sex difference between the make of the female cells and determined by the specific genetic factor is called the fertility factor RFR. When this factor is present in the F plus, that the make cells. If the factor is absent, F minus, it is the female cell. F plus is the male. If if factor is absent, F minus is the female cell. F factor exists two alternative states, either the part of the E. coli chromosome has the very small free circular chromosome that the multiple with the E cell division. The alternate case of the F plus cells potentially since the cannot transfer there the genes of only F factor is part of the chromosome can the female cell matter. F plus cells containing the integrated sex factor is called HFR, high frequency of recombination. Okay, F plus cells containing integrated sex factor is also called as HFR. HFR. The transfer of in this process, the transfer of DNA, the sex filly from the bridge between the F plus HFR F minus cells. Okay. F plus F. Okay, conjugation. Conjugation, especially these are the discovered in the Leeuwenberg and Tatum in E. coli 1946. Mainly transformation of the genetic material one bacteria to another bacteria. Transduction, another one is the transduction. Transfer of genetic material one bacterial cell to another. It means virus is known as the transduction. The bacteriophage, mainly they can involve in the bacteriophage. The transfer of genetic material from one bacterial cell to another means the virus is known as the transduction. The virus infects the bacteria is called bacteriophage. The phase, the transaction occur two ways, generalized and specialized. Generalized, the transaction is discovered. Ginder and Ledenberg in 1952, they found in the particular temperature phase act as the carrier bacterial genes from many different parts of the genome in the P22 and Salmonella specified transduction, especially mechanisms for the some of the bacterial phases. Life cycles of the bacteria. This is two types of the lytic and lysogenic. Lytic, the bacteriophage absorbs the bacterial surfaces and inject the nucleic acid into the bacterial cells. Inject the nucleic acids. Inside the bacterial cell, the viruses base multiples produce the several thousands of virus particles released by the lysis of the bacterial cell. These are the lytic cell. The lysogenic phase does not multiply in the entering the host cells. Lytic cycle also, number of cells are the multiplied in the internal part. Okay, already the surface injects the nucleic acid into the bacterial cell. Inside the bacterial cell, the virus or phase multiplies to produce the several thousands viruses particles which are released by the lysis of the bacterial cell. This is the lytic cell. Lysogenic is the lysogenic phase does not multiply on the entering the host cells. Instead of the viral nucleic acid integrates the bacterial chromosome. Now the phase is known as the prophase. When the lysogenic bacteria divides, the viral DNA also divides. When the prophase infects the another bacteria, only prophase are infect the another bacteria. It carries genes of the farm host. The phase is called as the transduction particle. Okay, transduction particle. In generalized transduction, the transducing phase can carry the 2 to 2.5% of the host chromosome. Only genes close to each other can be transduced together. In P22, the rate of transduction is about the once per every 105 infecting phases. The gene order the established two-factor or three-factor transduction by looking the number of various categories. The transduction. Okay, these are the transduction. And the next another topic is the genetic code. Genetic code. The central dogma molecular biology describes the two-steps process, which is the information gene flows into the protein. Describe the genetic information DNA transfer to RNA by process of the transcription. The RNA is responsible for the formation of the proteins by translation. Already we discussed the nucleic acid present in the AU. Nucleic acids present in the mRNA. AUCG are the arranged or organized in the certain codons which will have the information of formation of protein. The language nucleic sequence are mRNA is translated language, the amino acid sequence. The translation requires the genetic code to the which information is contained nucleic acid. Express the sequence of the amino acid. The collection of codons is also called as genetic code. Genetic code mainly, it is a set of the three nitrogen bases in mRNA. Set of the three nitrogen bases in mRNA. The set of three nitrogen bases in mRNA, which provides the genetic information for amino acid during the synthesis of the polypeptide chain. It defines uh, how triplet codons specify which the amino acid will be added next during the protein synthesis. Mainly, amino acids are what are the importance of the amino acid when it's a protein synthesis. And the term is given by the George Gamow. George Gamow. Genetic code, the term is given by the George Gamow. Actually, how many amino acids is there? 20 amino acids is there. At least 20 codons. 20 codons. What is only triplet codon? Each amino acid they give the triplet. Some of the codons are the only single codons. Some of the codons are the above the three codons. Only nucleotide acts the codon, if only nucleotide acts as the codon, there will be the 4 combination possible. That is the AUGC, which is not sufficient for the 20 amino acids. If 2 nucleotides acts codon, the also only 16 combination will be formed. Next one, actually the 3 nucleotide acts codon, there will be 64. 64. Okay, next, if three nucleotide acts codon, there will be 4 into 4, 464 combinations, 64 codons. According to Game, the postulated three-letter codon must be employed to encode 20 standard amino acid as living cells, the protein synthesis. These are the three codon, the AUGC. These are the especially three codon. What are the termination codons? Is also called as the termination codon, nonsense codon or stop codons. What are the actually 64 codons is there? Nonsense codon or stop codons or termination codons, the three. One is the UAA, Orki, UAG, Amber, UGA, Opal, Okri, Amber, Opal. These are the names of the UAA. These are the three are the stop codon. These are the three of the stop codon. How many? 64 minus 3 is the 61 codons is there. These are the three are the stop codons or nonsense codons or termination codon. What are the initiation codon? Initiation codons is the starting codon. Initiation codon is the AUG. This is the first codon of mRNA transcript or translated and codons, first amino acid protein synthesis. What is the first codon? Initiation codon is the AUG. AUG is also called as methionine. AUG is also called as methionine is the start initiation codon. Methionine is the initiation codon. The most common start of the codon is the AUG, and few of the mRNAs, GUG and UUG also act as the initiation codon. AUG codon is the unavailably some of the archaea bacteria. UUG and UUG is the acting as the initiation codon. Okay. In E. coli, AUG and UUG is the read the formal methionine, especially methionine. The AUG and UUG also act as the formal methionine, the initiation codon, is the used as the start codon. Similarly, GUG is the formal methionine, they act as the starter codon. GUG is also called as valent, read the codon regions. Okay, this is the what are the anticodons? See, initiation codon, termination codons, anticodons. The base sequence of the tRNA which pairs with the codons of mRNA during the translation is an anticodon. Codons written the 5' to 3 direction. Anticodon written by the 3' to 5'. Actually, codon written to the 5' to 3'. The anticodons also written to the 3' to 5'. The codons define which is anticodons to come next with the amino acid to create the protein strand. Anticodons help the bringing of the particular amino acid, bringing of the particular amino acids. That is the proper position during the translation. Actually, general codons are written part to the 5' to 3'. Okay. In the anticodons also they can start write the 3' to 5'. These are the anticodons. Anticodons also some of the tRNA molecules have pair with the more than one codon. Base number 123. The codon 5' to AUG 3'. Anticodon 3' to UAC 5'. Actually the codon is right to the 5' to 3'. Anticodon is right to the 3' to 5'. These are the anticodons and codons. These are the tRNA structure. These are the D loop, and next one the T loop. Next one is the anticodons are the located in the anticodon loop. These are the anticodon loop. Okay. These are the primary initiation codons also. These are the tRNA structure. These are the 5' and 3'. Okay. Anticodons also these are the located in the mRNA. Anticodons 3' to 5'. What are the characteristics of the genetic code? Triple code. uh, Triple code. The genetic code is the triple code consisting of the 64 codons. Each codon is the three-letter combination of the nucleotide. Actually, AUG. This one codon, AUG and GUG. And next one is the UAA, UGA. These are the one of the codons. Actually, these are the they can start with the three. Three is the codon. Three triplet codons consisting of the three-letter combination of the nucleotide. What are the characteristics of the triple codon genetic codes? 1 minute. Okay. What are the characters? Comma-less. These are very important. There is no comma. The gene is the transcript and translated from the fixed steering point, fixed point. Functions are not present in between the codons. No commas, no functions, and no a comma is less code as can be the written as UAE, UUE, CUC, GUA, UCC, ACC basis. What is the meaning of the comma-less codons? There is no comma in between the codons. Next results the drastic change in the genetic messages. No overlapping, comma-less, no overlapping. What is the meaning of the overlapping? For example, genetic code is the non-overlapping. Any single ribonucleotide specific location in mRNA pod of the only one triple codon. Each letter is read only once. Multilation changes in one letter would be the effect only one word. These are the overlapping of the codons. There is no overlapping. See, this one is the one codon to another codon. For example, AUG. U also they can code with the G. G also is a code. This is the overlapping process. Okay, no overlapping in genetic code. What are the characteristics of the genetic code? Is the comma-less, non-overlapping. Okay. The next one is the 1 minute. The next one is the genetic code is the universal. Universal. It means that is the same code used for the all life forms. For example, AUG. AUG is the universal. AUG is the methionine. The initiation codon. Okay. There is no not strictly true. There is the few context dependent of codons also contest. The genetic code is the nearly universal. Universal. These are the characteristics. What is the universal? Most of the organisms, UGA is the stop codon. CUG is the leonine. EAA, UAG is the stop codon. Tryptophan, mycoplasma, when AUG, these are the universal. Okay. It means the same code is used for the all life forms. For example, bacteria, some of the algal members, some of the animals. Next one is the some of the plant kingdoms. The universal codon. Next character is the unambiguous. The genetic code is non-ambiguous means one codon cannot specify more than one amino acid. Unambiguous. What is the meaning of the unambiguous? The genetic code is non-ambiguous. One codon cannot specify more than one amino acid. For example, triple UU code for the phenylalanine. It cannot code for the any other amino acid. Okay. However, ambiguous to at initiation have been encountered. AUG encodes for the methionine both in the internal sites of the internal position. Methionine is the modified into N-formyl methionine in prokaryotes. In prokaryotes, alternating integer codon is used. The GUG is common, and the specified valent internal site of the N-formyl is the interior position. See, these are the diagram is the um, only the no unambiguous. Mainly one codon cannot specify more than one codon. Okay, these are the another category. Next one, polarity of the genetic. What are the polarity of the genetic code? Mainly these are the prime to 3 prime. 3 prime is the end. The reading is from the right to left and left to right, then right to left. Okay. Next one is degenerate. Another character is the degenerate. What is the meaning of the degenerate? A given amino acid can be specified by more than one triple codon. Given these are the exception condition. The given amino acid can be specified by more than one triple codon. A collection of codon which specify the same amino acid is also called as codon family. The different codons of given amino acids are said to be the synonyms codons degenerates, degenerate residency, genetic code. The term is given by the Bernard and the Nirenberg. Degenerate is the given by the Nirenberg. These are the codons is degenerate, overlapping. The degenerates of the genetic code is the advantage of the organism when the two or more triplets with amino acid. These are the usually the third letter of the triple codon which differs. For example, arginine is the specified by six codons. The first two letters are the identical or four of these codons. Similarly, the first two letters are the same in remaining two ports. If the CGU codon for the arginine mutates to CGA, the letter codon also specify arginine. In fact, any base change in third position of any of four codons will specify the arginine. Thus, degeneracy of the code protective device against the effects of the mutations. Degenerate is the permit the number of silent mutations in which of the codon changes no amino acids. The proteins are the retains the normal amino acid sequence, and the codon mutation does not the results of the phenotypic changes. The Obbling phenomena, these are the Obbling phenomena according to Obble hypothesis. Third base of the codon is not very important, and the specify the codon is the particularly determined by the first two bases. Okay, it explains the degeneracy of the codon. The third codon, third letter. There is no important of the third letter. Obble phenomena. First two bases of the codon is the, for example, mRNA, the base pair that traditionally the second and third base is the anticodon to mRNA, 3' to 5'. But non-traditional base of the pairing observed between the third base of the codon, first base of the anticodon. The reduced specificity between the third base codon and the complementary nucleotide anticodon representing the Obbling phenomena. Oval position anticodon can pair with the C or U. U at the able position of the can pair of the A or G, adenine and the guanine. Hubble concept explains the sum of the tRNA molecules can pair with more than one codon. This means it does not need to 64 different tRNA molecules. Only half of the many are the required. Proline also 4 codon, CCU, triple C, CCA, CCG. The first three codons we recognize the single tRNA having the insulin of the first place. These are the genetic codon. Okay, these are the genetic codon. Thank you.